Good afternoon, everyone. My name is Elio, also from the University of Rio de Janeiro. And I would like to talk to you today about some molecular details of the p53 aggregation story. So I'll be focusing on this specific domain of the protein which undergoes the aggregation. Yes, you mentioned it's the DNA binding domain, but we call it the DVD to be simple. So in order to understand the aggregation of this domain, we have nature has provided some really great negative controls, and they come from the p53 family itself, p63 and p73, which these two proteins also contain DVDs, but interestingly enough, they show very low aggregation compared to p53. So this piece of data I'm showing you in the middle here is a classical aggregation assay using a molecule called bioflavin T, which specifically binds to amyloid. So essentially what you see over time under appropriate aggregation conditions, which is just 37 degrees Celsius, is increase in fluorescence of THT in p53, whereas p73 here in gray remains very low. So basically what I'm going to try to answer is the molecular basis for this discrepancy. And the tools that I will use are computational in silico approach of bioinformatics, sequence and structure analysis of the sequences, as well as molecular dynamic simulations. And then I'm going to present you various pieces of data and try to tie the bioinformatics and these simulation results together into an attempt to try to answer that question. Okay, so in this figure here, I'm showing you the domain architectures of the three proteins. You can see they're highly similar, and the region of highest similarity between the three proteins is their DNA binding domains, which is the focus of this talk. And below that architecture, I'm showing you a quantitative way to compare the sequences of the DVDs. In this the second one below, we look at the sequence comparison of 63 and 73. You can see that similarity score is quite high for their DVDs, indicating they are very similar. It turns out it's over 80% similarity in their DVD sequences. When you incorporate 53 into that similarity score, you can see still quite high values, but a little bit lower. So when you average that out, it comes to about 60% identity. So clearly the outlier there is p53, and essentially because the sequence of a protein dictates the structure, the answer is somewhere in here as to why the one aggregates and the other two don't. Of course, with this kind of high sequence similarity, the structures are highly similar. About only 30% identity would be sufficient to make the same fold, and we are over 60% in all cases. So the three DVD structures, which have been determined for some years, overlay quite nicely. Okay, so probably some of you are interested to know exactly the aggregation site in this DVD. It's highlighted here in red. And below that is the sequence of this aggregation prone segment for the three proteins. You can see there's a bunch of leucines and isoleucines in that sequence. And that's pretty typical for an amyloid-forming peptide, because what happens is these hydrophobic side chains will kind of intercalate and go together, excluding water at the interface, a classical hallmark of amyloid structure. You can see I highlighted a few differences between the sequences. However, when you pass all these three sequences through a bioinformatic tool to predict aggregation propensity, they all score extremely high, basically at 100%, indicating that there's a sure thing these are going to be aggregating sequences. So it looks like these small differences aren't the reason why the other two, 63 and 73, do not aggregate. p53 does. Of course, that would be too simple, and I have nothing more to say today. So I pushed on, and another thing you can do using the sequences alone is calculate something called the Instability Index. And essentially, this value, this parameter has a cutoff of about 40 for a protein being stable or unstable. So sort of something in the P53 sequence giving it this high value, whereas 63 and 73 have comparably lower values. And what this measurement came from a work from 1990 that found a good correlation between experimental protein stabilities and presence of certain dipeptides within the protein sequence. And so this value actually is an average of all of those separate dipeptides. And what I did was look at the dipeptides in 53 that had significantly higher instability indexes compared to the corresponding positions in 63, 73. Those are summarized here. And as I mentioned before, I'm going to show you some bits of data and try to tie them together a little later on, so I'll be coming back to that one. Okay, I'm just going to take a second to mention that although I'm talking about an in silico approach to this problem, in our group we have several people working on this in different aspects. So there's people doing cell work, animal work, and biophysical studies. And one of the tools we use at the university is NMR. Actually, the National NMR Center in Brazil is located at the University of Rio. So myself and other people do NMR studies to further investigate this story. And what I'm showing you here is just a proton nitrogen HSPC to P73. And we've actually assigned all of the proteins residences. And using this kind of thing, we can use recombinant proteins, put them together in NMR2, and look at the site-specific co-aggregation. As ESE mentioned, these proteins also can co-aggregate, and we can also look at the affinity of their associations. So it's very much, our group is multidisciplinary in incorporating these different techniques to answer the problem. So jumping back to what I'm going to talk about next, for the next couple slides, is the MD simulations that I've conducted on these DNA binding domains. So these are 2.5 microsecond trajectories that I performed on the 3 DVDs, and they were actually done in duplicate in order to ensure reproducibility and just gain more sampling. And of course, to have accurate simulations, I use explicit solvent. And it's worthwhile to mention that people have done simulations on these DVDs before in an attempt to answer this question. The answer clearly didn't come out yet, so one thing I decided was to do some extra long simulations. So these ones I'm showing today are about 100 times longer than previous reported simulations. And I think that's important because certain protein motions occur on obviously some longer time scales. And this is my idea. So obviously this is a pretty expensive computation, and I use the computing supercomputer clusters of the SharpNet network in Canada, and I have access to this through ongoing collaborators that I have there. So the next couple slides I'll show you some analysis of the MD results. One thing that you don't get in a static protein structure, which is the fluctuations over time, obviously. So one thing I wanted to look at was how the residue positions change from their initial structure throughout the simulation. I'm showing that for all 3 DVDs, the 2 replicates, and just because that's painful to look at, averaging those RMSD fluctuations out and plotting the averages here. You can see that overall there is some kind of consistent pattern among the 3. The regions that change in one are also usually changing in the others, but there's also some clear differences in this plot. And for the next 2 slides, I'm going to talk about 2 differences, one being this weird P73 peak here, and the second being the main focus, P53 regions of difference. So you can notice the region of difference here, a little one here, 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 and so on. So I'll show you that in some details. Okay, first off, this strange peak in P73, what's going on there, right? When I went to look at the corresponding residue position, turns out that this peak corresponds to an arginine. And I thought, okay, this is going to be, a different residue in 63 and 53. But it turns out they all have arginine in that position. Looking a little closer at the structure, what you find out is that this arginine is making a favorable electrostatic contact with distant glutamic acid in P53 and 63, and that corresponding position would be aspartic acid in P73. So just to summarize what those side chains actually look like, I have this little diagram here. So the main the one difference is that the glutamine side chain has an extra CH2 group. And immediately when I saw that, my idea was, okay, potentially this glutamic acid has a longer side chain, so it can make favorable contact, whereas the lack of that extra group makes the P73 incapable just due to distance constraints. So I decided to calculate the distance between the functional oxygen groups on the end of those side chains to the nitrogens on the arginine side chain, and average those out over time. What you see here is 53 and 63 have a distance under 0.5, which would be within the range to make a favorable electrostatic contact, whereas the nitrogens in the arginine and the oxygen and the aspartic acid for 73 are at a much further distance, where that electrostatic contact would really be quite negligible. Just to show you where this is occurring on the structure, it's in this region right here. In 53-63, you can see this kind of closed loop conformation, whereas in 73, it's kind of like this. So, although this was an interesting finding, I think it becomes more interesting when I found that these sites here are actually quite important for, or quite often mutated in P53. And when those positions are mutated, the stability in P53 goes down. However, we have this nice negative control, P73, despite not having this electrostatic contact, it still doesn't aggregate. So although this might be immediately an interesting position, it seems that it's not the the cause of P53 aggregation. However, just to show you another context of this, so this is experimental melting temperatures of the three DVDs. 53 has the lowest stability overall, 63 has the highest, and 73 is somewhere in the middle. Because the sequence of 63 and 73 are so close, this is basically one of the major differences in their sequence. This might be a reason why that P73 experimental stability is a little lower. At this point, we are making a mutant of the P73 to change that D to an E and do the thermal stability to see, just satisfy our interest to see if that is the case. So the second feature I wanted to point out in this RMSD plot are the areas where P53 differ from the other two proteins. There's several disjoint areas across the plot where that happens. Interestingly, when you show those on the DNA structure, they all kind of isolate along this lower half of the protein. Okay, so one other analysis I would like to show you has to do with hydrogen bonding. Just to quickly recap what a hydrogen bond is, I'm showing you a schematic of the protein backbone. We talked about the specific backbone hydrogen bonding. So hydrogen bond occurs between a proton donor, this amide, and the acceptor, which would be this oxygen. And it's hydrogen bonding is just determined by geometric criteria. So the hydrogen donor and acceptor distance has to be within a certain radius, and the angle made by the acceptor donor to the hydrogen has to be a certain angle for that hydrogen bond to occur. And these hydrogen bonds can be classified into two groups, one being protected, the other being exposed. And the distinguish between these two groups has to do with the number of nonpolar atoms in the vicinity of the hydrogen bond. The idea being, in the protected case, you have a lot of nonpolar groups around that hydrogen bond, creating somewhat of a watertight seal, which would prevent water from going in and attacking it. You have, under a certain threshold of nonpolar groups, you have this deprotected hydrogen bond, and potential for water to come in, which can also make favorable hydrogen bonds with these atoms, and attack that hydrogen bond, destabilizing the protein. And I think it's extremely interesting that if you run this analysis looking for exposed hydrogen bonds in the structures deposited in the PDB, which is the protein structure data bank, where structures usually get deposited, the one protein that comes out as the highest extent of exposed BHBs is prion protein, which is your classic amyloid-forming protein. So I performed this analysis on my trajectory frames, and what you see here is the result. P53 has this higher abundance of these exposed hydrogen bonds compared to 63 and 73. And I mapped those onto the structure here. Interestingly, they're kind of isolating on this bottom half of the protein. Okay, so I tried to combine the bioinformatics result with the MD results to hopefully get some idea of what's making 63 and 73 not aggregation prone compared to P53. So we know that those 63 and 73 are the negative controls, so they have some features in them that make them non-aggregation prone. So what I looked at was the sites that are protected in those two proteins for backbone hydrogen bonds versus P53, the regions with low RMSD changes compared to P53, and so on, basically for all of the criteria that I mentioned. And we're still working on incorporating other data into this. And kind of just cross-check between all of these things to identify single residue positions that are different between 63, 73, and P53, and the idea being those could be the important positions to that make them non-aggregation prone. But I've had some interesting results basically expanding off of, or growing off of these single residue positions to sterically proximal regions. And this model also includes identifying proximal residues that are complementary in charge. So their side chains have charge complementarity. And the idea being, like I said, these should indicate potential areas of interest that contribute to the lower aggregation of these proteins compared to P53. So I'll just show you one of these sterically connected pathways that I like to call them in this next slide. So here is the sterically connected pathway from P73, and you can see that this has a nice alternating charge, acidic, basic, acidic, basic, acidic, and you can tell based on this projection onto the structure that these are in quite good spatial proximity and they can form favorable electrostatic interactions. And the corresponding positions in P53 are shown here. In this kind of charge complementarity is abolished. It goes acidic, hydrophobic, basic, polar. And I've generated many of these kind of pathways. I'm showing you one here. But one thing I found promising from the result was that positions that are known to be mutatable to increase the stability of P53 came up quite frequently in the analysis. So the next step is to experimentally verify some of these things. So the idea is to mutate P53 positions into those of P63 or 73, and then test for aggregation propensity. And how am I doing for time? 3 minutes? Okay, since I have 3 minutes, I'm going to shift gears and show you a little bit different work, but basically what this other work involves is looking at this aggregation-prone region outside of the context of the entire protein, and it's also using MD simulations. And what I'm doing is simulating different numbers of copies, 2, 4, and some higher numbers in the simulation system together in order to see if and how they associate. And basically what I found is that they often associate in a typical structure that you might find for early oligomerization. And the applications for this would be determining, first of all, the mechanism of association, and drugs could be potentially developed to prevent that certain association. And in the future, I plan to incorporate in these simulations some specific compounds that are known to potentially prevent amyloid formation, a couple being resveratrol, a compound from grape skins, and obviously present in red wine, and curry spice curcumin. So that would be some future work, but for now what I can do with these qualitative plots here is build potential models of what a p53 fibril looks like. I think someone asked before if that's known, but it's not known. And I generated quickly some models based on these MD results, and also incorporating knowledge of how amyloid fibers should form. You get a classical left-handed helix along the fibril axis. Looking down, you have a water excluded cavity with those side chains associating quite nicely. So thank you for your attention, and I'm happy to answer any questions.
This lower region of P53, is that just an annoyance there to cause aggregation? Or there's a functional reason? Why is that area not more protected like 63 and 73? Do you know?
Sorry, you're talking about the aggregation prone?
Yes.
Side?
Why P53 in evolution didn't make that area protected like 63 and 73? This require this thing to be open for some functional reasons? Basically, if you make that thing rigid, P53 doesn't act, or do you have an idea?
I don't have any idea. I mean, you saw the sequence is quite similar among all the three proteins, and despite that, the other ones don't aggregate. The general idea is that P53, even in its wild type form, is prone to aggregation, and mutants, are enhancing the aggregation. These mutants that are enhancing aggregation are structurally destabilizing, so somehow this destabilization lets this region become very exposed.
Right, but what appears to be the case is this sort of this region to be more flexible, more accessible, is probably required for function. Otherwise, we expect the P53 during evolution will sort of get rid of it and try to protect itself, right? So just
yeah, I agree. There must be some evolutionary reason why it doesn't.
Anything else? Let's first thank Helio.